tinggal je. Tak sebab tak jadi.
Okay, so welcome to Imprinting Polymer. Okay, why this is important? Because in future, if you wish to do extraction, okay, a very specific extraction, if you want to retrieve from, let's say, you found a leaf, a plant, okay, that contain valuable compound in the plant, okay, and you really want to have a very pure, not to say very pure, at least a highly pure compound. Okay, this is one way that you can do. That's why it is called molecular sieve, because it can filter the molecule out, supposed to be specifically. But of course, because I did this for many years, there are some issues of selectivity, but it gives at least 70%, 80% purity, okay, which is actually quite good if you wish to extract, well, sometimes you need to extract a specific compound from that particular sample, okay? So why it is called imprinting, okay? That's the word, okay? So in chemistry, okay, let me move this one first. Okay, in chemistry, molecular imprinting is a technique to create templates, okay? Template shape, cavities. So this is the keyword, the template shape cavities in the polymer. Why is it moving? In the polymer matrix with memory of the template molecules. So if you look at here, so this is cavities. Yeah, so this is cavities. And we can see that this is what we call as templates. Yeah. So template means that we create these cavities specifically according to the shape of the template. Okay, so that is important for you to understand. The polymer contain cavities, okay, created. Yeah, that's why I mentioned just now that for this MIP, in short, yeah, it is called MIP, molecular imprinted polymer. It has to be specifically designed. So you cannot just buy a column C18, for example. Okay, you just get the column, the silica bit modified with certain functional groups. But for this particular imprinted polymer, it has to be designed. So this is the cavities inside the polymer, and it is designed to have the specific shape for this template. Okay, so how do we design, yeah, the cavities to have that specific shape of the template? Okay, probably you are not familiar. Okay, what is template? But at least you know this is cavities, and it's supposed to be a specific shape. So how does it? Okay, basically, in general, to design the polymer, okay, so this is template. Okay, you just take it as if we do like cookies with different shapes. So this is what you template is. You like the shape, the cookies to be in flower shapes or in different in star shape. So you have this what we call as template. All right, and then what we do with the during the polymerization process, okay, the template with, of course, the specific shape that you want, okay, is put inside a monomer solution. Okay, I hope that everybody still memory still can remember some of your polymer chemistry. So monomer, okay, because we are talking about polymer, so I hope that you remember what is monomer. Because I cannot go back, define back what is monomer. So let's say, so this template, okay, with specific shape is dissolved in a solution with monomers. It can be single monomer, it can be multiple monomers, okay? So in here, if you look at here, we have 3 monomers, A, B, and C. So I hope that you can recall back your polymerization. So what happened after that is there is interaction of the monomers. All these monomers will interact with the specific shape of the template. Okay, here there is binding process between the monomers and the template. I will show you the exact example, not the more realistic example after this. And then what we can do is, okay, this is the binding process. Okay, so once they have interacted, the monomer and the template, okay, so what we need to do now is to make sure that this process or this interaction remain as it is, remain there. You don't want them to detach, or you don't want the whole thing to uh, to be broken. You want the structure that we have created here to be solid, to be stay like the original interaction. So in order to do that, okay, these are also terms from polymer subject. Cross-linker. Yeah, what is cross-linker? So you have to do a bit of study if you don't know what is monomer, what is cross-linker. And of course, the next thing is the polymerization process. Okay. So in order to do polymerization, there are so many polymerization techniques that probably you have learned. They are radical polymerization, they are emulsion process. So depends on what kind of polymerization. You can also use radiation based polymerization. So that will be up to the suitability of the process. But basically, it should be interaction between the template and the monomer. We have to give time for the monomer and the template to interact first, okay, before we capture. I would say this to capture means you keep the whole thing in a solid phase. Okay, you can see this is the cross linker. So the cross linker is like you put a cage outside the template to make sure that it stays there. Yeah, the monomer stays there with the specific site interaction. And after that, what you do is to remove the template. Okay, you can see, so template here is, of course, if you are making cookies, you put the mold on the cookies mixture. And once you have got the shape of that cookies with similar shape with the mold, then you, of course, have to take the mold out. So once you have take the mold out, okay, you will have a polymer with cavities, yeah, with cavities that is specific to the original template. Yeah, you can see that this is supposed to be specific to the original templates. If you don't understand, please ask. Yeah, right. So So once you have done this, of course, as I mentioned just now, that it serves as molecular sieve. What does it mean? Meaning that the final polymer product that we can have from the imprinting process can be used to sieve a molecule from certain samples. So let's look at a proper example if we have after this. Okay, so this is the history of imprinting process to selectively capture various additives in silica matrix. Okay, all right. The process similar to molecular imprinting could be responsible for selectivity of antibodies. Okay, so again, this is there is another term for this imprinted polymer. It is called plastic antibody. Okay, why this called plastic antibody? Because, okay, let's just move. We'll show you after this. This is antibody system. Okay, so I think some of you who has taken sensor technology with me, we have discussed about antibody. Okay, so this is the shape of antibody, right? Antibody molecule. So antibody is a protein. And if you remember your basic science before, probably you learned in secondary schools that antigen-antibody interaction is very specific. Okay, so with recent pandemic, people know about a lot about the antigen test that you use for the rapid test kit. So it's actually an interaction between antibody with the coronavirus. So coronavirus is the antigen, whereas the antibody is the one that is fixed on the rapid test kit. Okay, so once you put your saliva on the rapid test kit, the antigen, which is the virus, will interact with the antibody specifically designed to capture the the coronavirus. So due to specificity of the imprinted polymer, so it works like antibody-antigen interaction. Okay, it's supposed to be very specific. It can be specific with a proper design of monomer interaction. Okay, so that's why it is called plastic antibody. Why it is called plastic antibody? Because if it is antibody, biological, original protein-based antibody, it has to be produced by inducing rabbits, mice, yeah, to get the antibody serum of the antibody. But for imprinted polymer, it's all synthetic based. You're using monomers, you're using polymerization, but it serves sort of the same principle. You have a specific interaction between the polymer and the molecules that you wish to extract or sieve. Yeah, so that's why it is called plastic antibody. Okay, so this is very important because if any of you have ever worked with antibody, it is a very expensive process to obtain the antibody. Okay, and because it is done in vivo, means that it has to be done using animals. They have batch to batch variation. Okay, the performance of the antibody can be different every time you harvest the antibody from the animals. So if we like switch to synthetic chemicals, right? It might be better because you can design, you can control the process rather than giving the animals by themselves to control the process. Okay, so this is very good for research, especially. You can be very good in designing and get a good imprinted polymer, which can be served as synthetic or plastic antibody. All right. Okay. Okay. Again, this is the history. All right. So what are the advantages? Yeah. What are the advantages of imprinted polymers? Target defines own recognition site. Okay, we'll see what do you mean by target? What do you mean by template? Stability, of course, because when we do like synthetic materials, it is more stable compared to biological. Okay. Specificity. Okay. Specific means that you can target certain molecules. Flexibility in use, of course. Facile means that easy to synthesize rather than you have to induce using animals. It can be used in non-aqueous media. Okay, this is also something very good. Disadvantages, okay. Diversity of binding site, poor processability, actually depends on the molecules that you are targeting, as well as a series of monomer that you have access to. Okay, so let's say now this is an actual process. Okay, so this is the template. Okay, so now you can see the real template. Okay, so the whole process is actually trying to produce an imprinted polymer to mimic an antibody, in this case, theophylline. Yeah. So this antibody binding site. This is the antigen. Yeah, this is the antigen. Okay. Sorry. So this is the antigen. So instead of using antibody to capture theophylline, we are using a polymer to capture theophylline. Okay. So how do we design a polymer? Okay. A polymer, okay, that is specific to theophylline. Okay. So what we can do, okay, please try to remember the last slide on the template just now. Okay, so this is the template. Okay. The one that you want to catch, the one that you want to sieve will become the template first. Okay. So you have to get a pure template. Okay. And then once you get the pure template, okay, we mix the template with the monomer. Okay. We will discuss later on how to choose the right monomer. Okay. Right. So this is very important. The process of choosing or predicting the right monomer. Okay, you can do the chemistry. Means that you can study the chemistry between the template and the monomer. You can try to predict the interactions, but now we can do AI, we can do computational to do this process, yeah, to match which monomer is the correct one. So you can use AI, you can use computational software to predict whether you have a good interaction or whether you will produce interaction, chemical interaction between the monomer and the template. Okay, so next step, of course, is the cross-linker, okay, because we want to cage them. We want to solidify and we want to make sure the interaction between theophylline, okay, and the monomer stays there. So this is what the purpose of polymerization. You have to use crosslinker, you have to use, if you are using radical polymerization, you need to have initiator. So that will be up to you. You can use other types of polymerization apart from radical polymerization. Okay, so next one, yeah, we have to cut this. Okay, if you look at here, the interaction is chemistry. It's a chemical interaction. It's not physical interaction. So you have to know, that's why you have to study the functional groups of the template. Okay, the one that you want to sieve, the one that you want to extract, the final, you have to know the functional groups. So you have to, okay, now you know you have ketone group, you have methyl groups, okay? So, and then you have the monomer with also a carboxyl groups. So you have to be aware of the functional groups of the templates as well as the monomer. Okay, once polymerized, so these are possible interaction. These are possible interaction that can happen. And for imprinted polymer, especially for those organic compounds, biological compounds, they rely a lot on hydrogen bond. Okay, so you can see we have hydrogen bond, okay, form between the template and the monomer. Okay, so now once it is interacted, as I mentioned just now, we want to capture them in a cage. We don't want the hydrogen bond to detach. Okay, hydrogen bond is actually a strong interaction. Okay, it's not actually as strong as covalent, but it's a very strong forces. Okay, so what you do after that, okay, in order to create these cavities, we have to take the theophylline template out. So in order to do that, we have to do a bit of extraction. So in this case, it's actually same thing that you learn. You have to use solvent, the right solvent to detach all this hydrogen bond. Okay. So once you detach this template, okay, now the next one, you have created a polymer with a specific cavities for theophylline. Okay, so in this case, if you like mix this polymer, the polymer can be in the form of emulsion, the final product, yeah. It can be in a form of emulsion, it can be in a form of beads, it can be in a form of you can solidify if you want to make it bigger. That's the beauty of this polymer. You can design it according to your needs. If you wish to be the polymer to be big, then you can you can make it into the size that you want. Okay, so when you mix it with a sample, let's say and you wish to take only theophylline from the sample, okay? So the theophylline is supposed to be the one that is being captured because this cavities is specific to theophylline. So if you have a mixture of other compound in that sample, it shouldn't be interacted with the polymer, okay? It should be the one that we design. So this is what we call as imprinted. Yeah? So we imprint the cavities with specific size, a specific shape, okay, of the molecules that we wish to extract. Okay, all right. So if you have imprinted polymer can be very good up to you can select a specific enantiomer. Okay, we know that drugs, okay, if any one of you work with drugs, different enantiomer can have different characteristic. One enantiomer can be a good drug, the other one can be toxic. Okay, so in order to separate, yeah, the enantiomer, so this is one way of doing it by using imprinted polymer. So this is example of L phenylalanine anilide. You have learned about enantiomers, yeah? I'm not good in organic chemistry, so I don't remember because I have left organic chemistry for quite some time. So you have this, I hope that you still remember the L, okay? So if you design the polymer well, you can separate the enantiomers, the correct enantiomers that you want to capture. So in this case, if we have L and D enantiomer, because you design it to catch the L, so the D enantiomer should not be in the cavities. So this is the importance of imprinted polymer. Okay. Okay, to understand how to choose, yeah, the monomer, because there are so many types of monomer. Yeah, there are so many types of monomer. So it is very important to understand what kind of interaction do you want. Yeah. When I say interaction, interaction means that this is not clear. Let's look at the previous one. Okay. The interaction between the monomer and the template. Okay. So we have to look at what are the possibility that can happen between this template and the monomer. So most of the time, as I mentioned, is hydrogen bond. Yeah, because why? Hydrogen bond is easy for you to remove or cut this, okay, and then you can easily rebind. But of course, it can be also covalent. Okay, it can be also covalent. So this is an example, covalent. But the problem with covalent, in order for you to extract, to reverse back the covalent bond is quite difficult. You have to use you have to use something difficult chemistry. But if you're using hydrogen bond, it's quite easy. So in order to use covalent, you have to make sure it is a reversible covalent linkage. If it is not reversible, then you cannot detach the template from the polymer. It's difficult to extract. Okay, so you have to choose reversible covalent between the template and the monomer. Okay, so this is an example of covalent interaction between galactose and the monomer. So this is a polymer form. This is a covalent interaction form between the galactose and the monomer. I don't know where, I can't see. You have so you have to break down covalent bond, which is quite difficult. Yeah. So in order to do that, it's a complicated chemistry to break a covalent bond, right? So I never did a covalent bond. It's always hydrogen bond, so it's quite easy. All right. So this is examples, yeah, a series of examples, templates, and the interaction with the monomer species based on covalent. Yeah? But for you to just expose yourself. Yeah. I have a book on imprinted polymer, but then it's quite difficult when we discuss about covalent. But probably some certain compounds requires covalent interaction. Okay. Advantages and disadvantages, covalent imprinting, ability to fix template in place. Of course, it is a very strong interaction. Yeah. Okay. Solvent means that it doesn't have to be, if it is a very strong corrosive sample, then it does not easily break. Can be difficult to remove template from polymer, okay, because of the covalent interaction. Limited number of chemistries for fixing template to polymer reversibility. Yeah, it's difficult. Yeah? Usually covalent is irreversible. All right. Okay, so what about non-covalent, which I used to? Okay, monomer template complexes. So you can see that most of the time is hydrogen bond. Okay, it can be also ionic, not ionic bond, is charges. Yeah, is the interaction between charges. You can see that this plus and this is minus. So there is no electron transfer. It is not ionic bond, but it has charges interaction, the positive and negative charges interaction. So this also can be interaction between the monomer and the template. So this is pseudo, it's not pseudo. It is still also interaction because M represent less electron and nitrogen rich electron. So there is an interaction between these two. Again, so all right. These are also examples, more examples on non-covalent interaction. Okay, so these are examples of the template, types of template that you can use for non-covalent. You can see the covalent just now is more on organic compound. So this is acid bases with charges. Yeah, acid bases, they have positive, they have acids like bases, they have negative charges. Acid have positive charges. Okay, so disadvantage and advantages, easy to remove template from polymer. Okay. Rapid kinetic, okay, for rebinding of the template. Inability to fix template in place during polymerization. Of course, because when we talk about hydrogen bond, interaction of positive and negative charge is not that strong, but then, yeah, it's still useful. Generally requires low polarity aprotic solvents. Okay, that's the biggest issue. If your sample is in water, because water is polar, then sometimes it's going to be difficult. Because we understand if the interaction is hydrogen bond, and if the sample is aqueous, water, there is also hydrogen bond, then there is competition between the solvent, which is water, with the interaction of the polymer and the monomer and the template. Okay. Okay. Also, there is semi-covalent. All right. We skip this. You can do spacer. Spacer means that you, instead of just monomer, we can have an extended spacer. It's like you make it long for the long hand so that it can interact with the template. Okay. Right. All right. Target molecules as well. It can be difficult if the molecules that you are targeting is very small. Okay, it can be very difficult. If you are targeting big molecules, then it is quite easy. Okay, so you can see that the target molecules, there's a lot of variety. You can target pesticides, you can target amino acids, drugs, protein. So in terms of application for imprinted polymer, there's a lot that you can do to extract the molecules of interest by using imprinted polymer. Okay, this is the most important part, okay, before we, right. When we are to design imprinted polymer, so these are the things, very important things to decide, okay? Monomer, porogen, so these are all the terms that you have to get used to. What is porogen? What is solvent? What is initiator? Initiator, if we choose radical polymerization. And types of cross-linker, reaction temperature. Okay, and time is up to it can be 48 hours, it can be 24 hours, okay? The final physical form of MIP is solid, okay? But then you can also use different types of polymerization so that you have emulsion, okay? So you don't have to choose a monolith. Monolith means that it's a solid form, okay? Because finally you can have different form of different sizes of polymer that you wish. If you choose radical polymerization and you end up with a monolith, is a bulky hard polymer, you have to grind it, yeah? And then to make sure it's to the size that you need, okay? But you can also do like coated silica particles, meaning that, what does it mean? We can polymerize on top of silica. So silica is like the support for the imprinted polymer. Okay, so this probably you cannot picture, but depends on the final form that you want. What final physical form of the polymer, imprinted polymer that you want. So we can design it. Okay. It can also be in a form of membrane. So you can put the polymer in a membrane. So if you like want to filter water or any sample in liquid form, so you can put the imprinted polymer particles in a membrane, and when you flush through the liquid samples, then you can capture the molecules of interest in the membrane. But it's quite difficult. I've done that and it was not successful. Yeah. That's the beauty of the polymer. You can design the final form in what kind of final form that you wish to have. Okay. This is one type of polymerization. If you choose precipitation polymerization, you have emulsion. You have a very small size of imprinted polymer, and is in a form of slurry. Okay, so it can be in that way, but when you have that, it's quite difficult to separate. Then you have slurry. Yeah, emulsion polymerization. Okay, let's. Okay, so this is the most important application of chromatography of imprinted polymer. It is designed to be the column of the chromatography if you wish to separate specific molecules. Okay, if you want to separate specific molecules, so this imprinted polymer can be the column package, column pack material. Yeah, right. Okay, this is how it looks like. You can put the imprinted polymer in a column, okay? And remember the video that I have sent you last week, okay? So those are examples, yeah? So I will try to, well, I cannot show it here. I don't have the video inside the laptop, okay? And as I mentioned at the beginning of the class just now, that it can become a plastic antibody. So, but it's actually difficult. You have to be trained, yeah. So it can be an antibody, plastic antibody, instead of antibody that is obtained from animals. All right, so this is antibodies from animals, and this is MIP. This is the comparison. So I think I have discussed the comparison just now. Okay, I'm not going to talk about the sensor. Let's skip this. All right, so I think we stop now, okay? This is going to be the second session. So I'm going to present the work that we have done using imprinted polymer, okay? So this is an example of how the polymer is prepared, and particularly the design is for specifically targeting penicillin. In this case, penicillin G. There are a lot of different types of penicillin-based antibiotic. So this is our project on the imprinted polymer for penicillin. So we will see you. I'll see you at 8 o'clock. Any questions? Anybody?
No, Prof.
All right. So welcome back, everybody. Okay. Another 2 minutes, right? So this is the application of imprinted polymer, one of it, okay? So in this case, it is being used as what we call as receptor, yeah? Chemical receptor in order to detect penicillin G. Yeah, penicillin, there's a wide variety of penicillin. So one of it is called penicillin G, pen G. Okay. So let's look at why it is important to detect any cross contamination because in pharmaceutical company, the production of non-penicillin drugs and penicillin drugs has to be properly separated. Yeah. But unfortunately, sometimes due to people, you know, going here and there, so it's difficult. It is a real problem. Sometimes there is cross-contamination of trace residue of penicillin in the non-penicillin drugs. Okay, so this is also possible, and there is also because some antibiotics are used in kettles, so there are residues of antibiotics in milk. So it is also sometimes important to check whether there is residues of penicillin based antibiotics in milk, some food, some milk based food. Okay, because some people are allergic, yeah, allergic to penicillin based antibiotics. Okay, you can see there's a possibility of pressures, and worst case, it can be anaphylaxis, yeah, anaphylaxis. So, and this one is a serious matter. You can die from this unless treated. Okay. So, if we look at penicillin in general, not just penicillin G means that the side chains are different. The most important is the beta-lactam ring. Okay, so this represent the penicillin, the what we call the unique penicillin chemical structure, which is the beta-lactam ring. So in this project, we are to design imprinted polymer that is specifically detecting the beta-lactam ring, which is quite difficult. Okay, so we start with of course, as I have discussed before, it is very important to decide on the monomer. Okay, so in this case, the template is penicillin G. Okay, this is penicillin G, and we have to decide the best monomer. Okay, to uh, perform the product, to fabricate the polymer, yeah, the imprinted polymer. So here we have penicillin G, and the monomer chosen is acrylic acid, MAA, and this is the cross-linker. EDGMA is an example of cross-linker. Yeah. So this is an example of cross-linker, and this is an example of monomer. Okay, the monomer, of course, you have to choose based on the functional groups of the templates, and we can also do computational study, as I mentioned earlier, as well as AI. Yeah, AI software, same goes to, okay, you can select the interaction, the best interaction, the best binding, and then you can choose the monomer. So in this case, there are many types of polymerization that you can choose. So, but here we used all phase emulsification polymerization. So this is among the steps. Okay. Of course, the thermal or the temperature must be controlled. So all this has to be studied as well. What is the best temperature? How long the polymerization? You can see that it is in fact quite long hours. And then the next one, once we have the polymer ready, is the extraction, the removal of the template. This is also critical, and we have to study what kind of solvent system that is suitable to remove the template, in this case PNG, from the imprinted ready-made polymer, a fabricated polymer, prepared polymer. So starting from the monomer, you have to study, and the temperature and all other controlling factor, porogen. Okay, I have not talked about the porogen. The washing to remove the template. Okay, so porogen is here. Porogen, what is porogen? Is actually solvent. Okay, because whenever that you want to do polymerization, all this, okay, all this component must be dissolved in solvent. Okay, so it is called porogen for some reason. I will show you why it is called porogen. So it's very important, because if you don't have the right solvent to dissolve all this, then the polymerization will not happen. Okay, you cannot have a homogeneous polymerization. So these are all parameters and factors that you have to study if you want to prepare imprinted polymer. Okay, you can see from here, the most important, one of the most important is to study the ratio. When preparing imprinted polymer, we have to study the best ratio of monomer, template, which is the PNG in this case, and the crosslinker. So this one also we have to study a series of ratios, and then in order to have the best physical form, one thing, and the best characteristic of the imprinted polymer. This will take some time. The study of the ratio, the formulations is quite tedious. Okay, the next one is to check, okay, whether the imprinting process happen or it does not, whether it is not successful. So we have to check. So in order to do that, we polymerize a control. So this is what we call as non-imprinted polymer. Means that you polymerize, okay, all this EDGMA and the monomer, we polymerize without the template. Okay, so it's like empty polymer. Okay, so once you polymerize, and then we do, first thing is to look at the FTIR. Okay, it's quite difficult. You can see not much difference. Usually what we see is only a shifting of certain peaks, broadening of certain peaks, or shifting of certain peaks. So it's quite difficult. But most of the time, if we are to look at hydrogen bond, to observe hydrogen bond, we will look at this area. Okay, if you have a broad band here, there is a possibility of hydrogen bond is formed, meaning that there is interaction between the monomer and the template. Okay, so the first thing to look at, and the most important, is FTIR. All right, so when we talk about porogen just now, so the purpose of having a porogen, the right solvent to prepare the polymer, is we need to create porous. Yeah, we need to create porous surface of the imprinted polymer. Why? Because that porosity will allow the rebinding of the PNG in this case, because we want to rebind the PNG from the drug. So if there is no porosity means tiny holes on the surface of the polymer, okay? So if there is no porosity, then the polymer will not be able to rebind the molecules that we designed to rebind. So it is important to have a good solvent, which is what we call as porogen, okay, to get the best porosity. So from FISM EDX, we can see the distribution of these tiny particles because it's a nano emulsion polymer, okay? And then from the EDX, we can see some, if any, difference in terms of elemental, in terms of percentage. So I'm not going to talk about the detail of the results. Okay, so this is highlights of the results. You can see some porosity, but the best porosity is through BET analysis. I'm not sure whether we have it or not, but this is the most important. Okay, the imprinted polymer just now is designed to pick up PENG. So if you look at here, we also test with ampicillin, which is penicillin based as well. And thiamphenicol is non-penicillin. So how do we know? You do not see any lactam ring here. So ampicillin is penicillin based and there is lactam ring. And this is PENG, lactam ring also. So thiamphenicol is non-penicillin antibiotic. And we need to compare also with non-imprinted polymer. So the one that PENG, we can see a higher rebinding, okay, compared to the ampicillin and thiamphenicol, because these are not the one that we designed the imprinted polymer for. The imprinted polymer is supposed to have a specific shape for PENG, so that is why it's supposed to pick up PENG. But unfortunately, it is not 100% selective. You can still see there are some rebinding of other types of antibiotics, but that happens as well. Okay, ratio study just now is very important. And one thing we need to look at, because if you have a smaller size of nano emulsion, meaning that it has more surface area, then it can absorb more during the rebinding process. So these are among the things that we look at. And this is the physical form, yeah? This is the physical form of the MIP. So you can see the PENG is inside here before extraction. Means that you have not taken out the PENG as the template. And this is the non-imprinted. There is no PENG inside, there is no template. And this is after extraction. So it's difficult to have 100% of the PENG removed from the original MIP. So this is the final product. Still, you see the yellow color because it's difficult to extract 100% from the imprinted polymer, the PENG from the imprinted polymer. Again, because this is research-based, showing evidence of the nano sizes are very important. So using FISM again to evidence the, sorry, let me move back, showing the sizes, yeah? Right, next one. So I'm going to show you another type of imprinted polymer. In case we are to remove mercury. So just now we are to design imprinted polymer for penicillin. The purpose is to build a detection system for penicillin. Okay, but this one is basically to remove mercury from water and also oil and gas waste. All right, so you can see this is just now penicillin is beta-lactam and is organic, right? Is the functional groups organic, okay? But this time around is ion, okay? Because mercury is in the form of ions. Okay, so it's very important to understand what types of monomer to be used, yeah, in order to get the best interaction between the ions and the monomer. So sometimes you have to add additional. So in this case, it's not just monomer. We also add ligand, okay, because this ligand is very has high affinity towards mercury, because mercury loves sulfur. Okay, so in order to like have better rebinding with mercury, instead of just monomer, we add some more additional of polymerizable ligand. Yeah? So in the end, you can see there is a bit of complex structure, and same procedures. We do polymerization and then remove the template. So by right, we have should have a specific site for mercury ion. As you can see, mercury ion is smaller size compared to the penicillin G. So it's actually quite challenging to design imprinted polymer for smaller and smaller ions or smaller protein. Yeah? So this one is quite difficult. But you can see from here, just now we have emulsion, yeah, very small particles. But as I mentioned in the earlier, the first hour, that you can design the final physical form. How you want to look to make it look like. Okay, you want it to be big, you want it to be in membrane form, you want it to be in smaller particle size, depends on the application. So in easier to be because we are talking about removing mercury from water. So we need to have something to be easy to filter. Yeah, if it is too small, then we have problem with filtration system. So we do sizes like cylinder size because it's quite easy to filter. And this is the whole process. You can see that this is the same process that I have shown just now. And this is the final product. Again, we use FTIR to check whether we have the MIP process is successful or not. So we compare with the monomer, IR of the monomer, and then IR of the imprinted polymer. So there is a band broadening of here showing that there is a possibility of hydrogen bond, yeah, because this is what we expect to happen. There is interaction between the sulfur and the mercury. And again, we need to study the porogen. We look at the porosity. You can see that different use of solvent, different ratio of solvent, give different types of porosity. Okay, so the best porosity we need, you can see that this is not porous. Yeah, so this is porous and some are a little bit porous. Okay, why we need this porosity? As I mentioned just now, we need to rebind the mercury from the water, for example. If it is solid like this, then it's difficult for the mercury to be inside the polymer. So study on the porogen is very important. Sometimes we have to use a mixture of the solvent as the porogen, not just a single solvent, because that will give, you can see a better results in terms of surface area. We have to study all this instead of just using single solvent. And these are all the selectivity. These are all the study on the rebinding. So here we can see that imprinted polymer is designed for mercury. So when exposed to different metal ions, it's supposed to be low, and for mercury is supposed to be high. Okay. Right. So this is an example of column. Okay, where we put the imprinted polymer. And this is the waste with mercury, and this is where it moves, and then the mercury is supposed to be trapped here. And then this is the clean water after treatment of mercury. Right, so these are all application on sensor, because my research is on sensor, so the application is most of the time is sensor. So I'm not going to like discuss all this because you're not taking sensor. All right, but there's a lot of application on imprinted polymer in sensor. All right, so this one we did it before for the workshop. So these are examples of what we have done, and that's it. Any questions? Because we are going to finish early. Any question on imprinted polymer or any other things related to my lecture? Okay, for your final exam, please focus on solid phase extraction. Yeah. So as I mentioned during the last week lecture, you're not going to have any calculation-based questions, because I think you have done it during your undergraduates probably. So no calculation, but more descriptive, yeah, on solid phase extraction. And I'm not sure whether I have to give you assignment or not. I have to check with Dr. Sazinda because I only spent 6 hours. I don't know. So I have to check. So any questions? All right. So if there is no, thank you so much. So my lecture is until here. And next week, I think Dr. Sazinda have a plan for you on invited lecture. Okay, thank you, everybody.
Thank you, Prof.
Right.
Thank you, Prof.
Thank you, Prof.
Thank you.